Thank you so much for having me. I'm really happy to be here to discuss RCA's role in monitoring the Rivanna watershed and some of the different organisms that we collect to help us determine the health of local waterways. Oh, here we go. Hello. So I'm going to leave mine on for a little bit, but I have a lot of photos and information that I don't want to cover up. So at some point I'll turn off my video so you can see my full slides. So yes, I am the monitoring program manager for the Rivanna Conservation Alliance, which is a small nonprofit watershed organization based in Charlottesville, Virginia, but we work all over the Rivanna watershed. And we were formed in 2016 by a merger of two different organizations, one of which was the Rivanna Conservation Society and the other was Streamwatch. I'm gonna go ahead and stop my video. And our mission is to work with the community to conserve the Rivanna River and its tributaries through monitoring, restoration, education, and advocacy. We have a lot of different programs at RCA, but by far the biggest thing that we do is water quality monitoring. And we currently have two different volunteer community-based water quality monitoring programs. We have a bacteria and a biological monitoring program, both of which are certified at level 3 by the Virginia Department of Environmental Quality, which is the highest level of certification that citizen monitoring groups can reach in Virginia. And this means that our data is considered to be the same quality as DEQ's, and that DEQ can use our data as if it was collected by the agency itself. And it also means that RCA's data can be used by local, state, and federal government agencies for environmental decision-making. So it's very, very useful and has a wide-reaching impact. And RCA has the unique distinction of being the only nonprofit organization in Virginia to have level 3 certification for our bacteria and biological monitoring programs. Our biological monitoring program has been around for about 18 years now, so quite some time. And at most of our long-term monitoring locations, we have data that goes back to about 2003. So for this program, volunteers and staff go out and sample stream organisms called Benthic Macroinvertebrates at 50 long-term sites that we sample each spring and each fall. And our spring monitoring season just started this past Monday, March 15th. And for this program, volunteers are trained on the sample collection and organism identification. And at the bottom of this screen here are two examples of organisms that we collect. The one on the left is a stonefly nymph, and the one on the right is a dragonfly nymph. And we're going to look at a lot more organisms that we collect in a couple of slides. So what are benthic macroinvertebrates? Well, benthic means bottom dwelling. So these are organisms that inhabit the stream bottom or river bottom or the substrate. Macro means that they are large enough to be seen with the naked eye. So you might need the aid of a microscope or other tools to identify the organisms down to the taxonomic level that you're interested in, such as the family level or the genus or species level, but you can observe them at least with your naked eye. And invertebrates means these are animals without a backbone. So they might have an exoskeleton that covers their entire body or portions of their body, or they might be completely soft-bodied. And benthic macroinvertebrates are a group that includes insects, crustaceans, mussels, clams, worms, and all of which spend at least some portion, if not all of their lives, in aquatic environments. And they are a really important component of freshwater ecosystems. They feed on detritus and plants and have a major influence on nutrient cycling in ecosystems. And they're also a really important food source for other aquatic and terrestrial animals, including fish, birds, and amphibians. So why benthic macroinvertebrates? Why do we sample those organisms? Well, they are considered biological indicators and are reflective of environmental conditions. So in this case, these organisms are reflective of stream health because they vary in their sensitivity to or their tolerance of different types of pollution. So they've actually been studied for over 100 years now in terms of their pollution tolerance or sensitivity. And the organisms have been assigned a pollution tolerance value based on a scale that ranges from 0 to 10. So organisms that are really, really sensitive to different types of pollution would fall on the lower end of the scale. So anywhere from like 0 to 4. And then we also have some mid-range organisms that are moderately tolerant or moderately sensitive to different types of pollution. And then we have really, really tough bugs that are able to withstand stressful conditions. So they would be on the higher end of that scale, anywhere from like a 6 to 10. They're also really useful to study because they're extremely widespread and they are found in all types of aquatic environments all over the world. They're also super useful because they have a limited mobility. So on average, they will spend 1 to 3 years in the stream or river in their immature phase as either larvae or nymphs, depending on which organism you're talking about, before they actually leave the stream as flying insects as adults. And biological monitoring is super useful because it gives a longer term picture of water quality as opposed to going out with like a monitoring sonde and collecting temperature, pH, or dissolved oxygen, which is definitely very useful, but it can change, hour to hour or even minute to minute. So biological monitoring is super useful because it gives you a longer term sense of what the overall water quality is at a given site. This slide shows just some of the organisms that we collect when we go out to sample at our locations. And this is a simplified breakdown of where the organisms fall on the pollution tolerance or sensitivity scale. So on the left side here, we have really, really sensitive organisms that are not able to withstand certain stressful environments or streams or rivers that have high temperatures, low dissolved oxygen, lots of sediment inputs, pollutants such as nutrients or other stressors. And so some of these organisms that are really, really pollution sensitive include things like stoneflies, mayflies, case-making caddisflies, which are a really, really cool group of organisms. So this photo on the left shows a caddisfly larva, and they're pretty cool. The case makers make these little cases out of little materials that they find in the stream. So depending on which family you're looking at, a case maker can make its case out of little bits of twigs, little broken up pieces of leaves, small bits of gravel, or even little bits of sand. So it's pretty cool, these little homes they make for themselves. And this category also includes gilled snails that require a lot of dissolved oxygen in the stream in order to survive. And these organisms on the left side also typically have visible gills that they use to breathe. So you can imagine that, they wouldn't do quite so well in streams that have a lot of low DO. Then we have like a mid-range category, and these include organisms that are somewhat sensitive or somewhat tolerant of pollution or stressful conditions. And these are organisms that include dragonfly nymphs, damselfly nymphs, clams and mussels, crane flies, crustaceans such as sow-bugs, scuds, and crawfish or crayfish, depending on who you talk to. Hellgrammites are also in this category. And a particular family of a caddisfly is in this category, and they are separated out into this kind of mid-range category because they are less sensitive than the other caddisflies that we study. This is a family called the net-spitting caddisfly that tends to be more tolerant of nutrient pollution. So that's why they're in this particular category. We also collect beetles in our streams, and beetles are generally somewhat sensitive or somewhat tolerant of pollution. One family that we study is the riffle beetle at the very bottom, and they are one of the few organisms that we collect that actually complete their life cycle in the stream. So picture here at the bottom is the larval stage and then the adult stage. And then kind of our final category is our tolerant category. And these organisms that are pictured here are not super attractive per se, and they, just by looking at them, you can probably tell that they're able to withstand stressful environments because their bodies are so simplified. So these are organisms that are able to withstand generally warmer temperatures, lower dissolved oxygen. Some of them are pretty tolerant of streams that have sediment issues or problems with organic pollutants. So some of the organisms that fall under this category are organisms such as the midgefly larva, blackfly larva, leeches. Thank goodness we don't really collect leeches. They tend to prefer slow or still waters, and we sample in running waters. So we sample streams and rivers. So we don't typically find leeches in our samples, thank goodness, but they would be considered a tolerant organism. We do also collect worms and flatworms, and they are also tolerant. And then there's a different type of snail that falls into this category called the lung snail, and they are very tolerant of low oxygen. So I'm not going to go into a lot of anatomy on this slide, but I just wanted to generally compare two different organisms, one of which is the mayfly nymph, and pictured here is a beautiful, beautiful flat-headed mayfly, the family Heptageniidae. And they, just by looking at it compared to the midgefly up there in the corner, is a lot more complex compared to the midge. So they have a lot more specialized areas and body parts compared to the midge. So attached to the abdomen here are these like feathery gills, which hopefully you can see against this background here. So that's how they obtain their oxygen, is right by absorbing it through their gills here attached to the abdomen. And then the midge, they do not have gills. They actually obtain their oxygen in a different way. It's by a mechanism called cutaneous respiration. And that just means they have the ability to absorb oxygen all over the surface of their body, rather than having to rely on a really small, specialized, like localized area to absorb dissolved oxygen. So you could probably imagine that in a stream with low dissolved oxygen, this mayfly probably wouldn't do so well. Probably have to work a lot harder to breathe and to do well and thrive in that kind of environment. So I thought it would be kind of neat to compare two different rural streams that we sample in Albemarle County. We have we have a station on Doyle's River, which is a super beautiful, like really, really nice stream, a really, really healthy stream. And then we also sample at Stockton Creek, which is not so healthy. And so just by looking at these two pictures, you can notice a lot of differences. In Doyle's River, you can see that the water is connected to the floodplain. It's kind of hard to see with the shadows, but there's a really excellent substrate here. There's a diversity of cobble sizes, boulders, different little pebbles and rocks. There's a little bit of fine sediment here, but not so much sand or silt. And so the diversity of cobble sizes really provides a lot of different habitats for these little organisms to live. And everyone has their niche in the stream. So this healthier stream is able to support more diversity of aquatic life as compared to Stockton Creek. And you can see at Doyle's River, there's plenty of riparian vegetation that would help naturally filter any runoff that were to enter Doyle's River. Doyle's River is also really shaded, provides a lot of canopy cover, and which also cools the stream and keeps temperatures cooler. Stockton Creek is a little bit of a different story here. You can see that the banks are super high, really, really incised banks. It's actually kind of hard to access this stream in certain locations because the banks are so high. And you can see all that exposed sediment there, which you can probably imagine after a big flood event or during a scouring event. A lot of sediment does end up in Stockton Creek. And so the substrate at Stockton Creek is not as nice as Doyle's River. It's pretty flooded with fine sediment. And you can see that there's basically no canopy cover in this little section of the river. So there's no like real stream shading. And this is a stream that definitely has higher temperatures. So you can probably imagine that mayfly on the previous slide would have a harder time surviving in Stockton Creek compared to Doyle's River. and that, midges, it's not that we don't find, tolerant organisms in healthy streams, but healthy streams have well-balanced populations and have lower relative abundances of tolerant organisms. So that, Stockton Creek likely has a lot of midges in it. And we're actually going to compare recent data that was collected at both of those locations. So each time that we go out and sample, we collect our organisms, and each sampler produces a final stream condition index score that ranges from 0 to 100. And if the stream score is below 60, that means that the stream is considered impaired, meaning that it doesn't meet Virginia's water quality standard for aquatic life use. So it means that it's a pretty unhealthy stream if it falls below that 60 impairment threshold. So these are some recent monitoring results from Doyle's River that were collected in spring 2019. And each piece of this pie represents a unique organism. And so we collected 19 unique organisms or total taxa from this sample, which is pretty good. It's pretty diverse. And most of the organisms that were collected in this sample are considered sensitive. And then about 20% were considered to be tolerant. So this is a pretty well-balanced population here. Mostly sensitive species were found. So this would have gotten a very good rating in our assessment. And then comparing to Stockton Creek, which doesn't look so good, we see that about half the sample comprised tolerant organisms and about the other half was sensitive. So it's not the worst case scenario. It's not the worst we've seen, but it's definitely impaired. It fell, beneath 60, so it would be rated as impaired. And the rating that we would give it would be a fair. It's not as diverse as Doyle's River, which is probably not surprising. Each year we publish a stream health report that summarizes 3 years or 6 seasons worth of data that we've collected from our biological monitoring program. So this is a map that shows the ratings based on data collected from 2017 through 2019. And you can see that about 68% of the streams that were sampled during this time period fell beneath that threshold value of 60. So they were either rated as fair or poor, meaning that those streams failed to meet Virginia's water quality standard for aquatic life. So a large portion of the streams, did fall in those two categories, and it's pretty common for sites to, fall from good to fair or even go the opposite way. But we did have an overall downward trend from the previous report. We did have 7 sites that changed from good to fair, and then 2 sites that changed from fair to good since the last report. And so that's an overall downward trend, which was likely due to the record amount of rainfall and that the watershed received in 2018. And we also had a lot of high flows and scouring events that occurred in 2018 and 2019. So that probably has a lot to do with this downward trend that we saw. So while it's really important to look at these short-term shifts in stream health, it's important to also do periodic assessments of long-term trends in benthic scores. And so something that we did recently was a long-term benthic trends analysis using 15 plus years worth of data that we've collected from our organization. And so we used a data set that comprised of over 1,000 fall and spring samples that our organization has collected. And we did detect some long-term improvements at 4 different locations. And those are the ones that are listed here: Buck Island Creek, Meadow and Morris Creek, and then Rivanna at Milton. And all of these sites are located downstream of large-scale water quality improvement projects, such as BMP installations, stream restoration projects, and then upgrades to a large wastewater treatment plant. This would be the one on Morris Creek that's operated by the Rivanna Water and Sewer Authority. So that was kind of an exciting find. Although we did detect one decline in Marsh Run, which is a heavily incised stream, has really, really high banks. It's very clear that a lot of erosion happens there during high rainfall and storm events. And so it could be that excessive sedimentation could be contributing to declining scores here. And so we're going to keep an eye on it and continue to monitor there. Although it's important to note that we do have a shorter time period at Marsh Run. We only have about 9 years worth of data there compared to 15 years at the other locations. So we're going to keep monitoring there and keep doing this long-term trends analysis every 3 years to keep reassessing over time how things are looking. And if you're interested in reading the full report, I provided the link there on the slide if you want to check it out and read more about the improvement projects that are located above those improvements that we saw. I do want to mention a pretty cool project that our monitoring program completed in collaboration with the Virginia the Virginia Department of Wildlife Resources in 2019. So in the summer and fall of 2019, we went out and surveyed 24 different locations in the Rivanna watershed, most of which were in Albemarle, and we collected fish data. We ended up collecting 56 species, which is considered very diverse, and 44 of those were native, 12 were introduced, and 2 were migratory species, which included the American eel and the sea lamprey. We did collect a lot of sensitive species, such as the long-nosed dace, the northern hogsucker, rosy-side dace, and a few different darter species. And then we did collect a couple of keystone species, which are species that other species in the ecosystem largely depend upon, one of which is the bluehead chub, which is pictured here. And during the reproduction or spawning season, the males construct these large gravel mounds or nests in runs and glides that other minnow species use as spawning grounds. So they aren't able to do this in streams or rivers that have a lot of fine sediment because they need a specific substrate or cobble size to be able to construct these mounds. So excessive sedimentation can have a very negative impact on the species and those species that rely on them. And in Virginia, at least 13 different species, including the mountain red-bellied dace, which is another important sensitive and keystone species, rely on these mounds to reproduce. So they're a really, really important keystone species and have a big role in the overall ecosystem. And I don't have that much time to talk about a lot of the species that were collected in great detail. But if you're really interested in fish or just are curious about what we collected, I would encourage you to follow this link, rivannariver.org/fish, for some more updates on our fish-related work. And you'll also find a link to an interactive story map webpage that goes over some key highlights and findings from the survey that was conducted with DWR. And it's a pretty fun website to work through and a pretty fun read. So I would encourage you to check that out. So we share our monitoring data with a lot of different people and organizations. We share our data each year with our community partners. So we do publish a stream health report each year, and that gets sent around to local governments and community partners for their own purposes and decision-making processes. And we also provide all of our data on our webpage if you're interested in previous data. And then each year we also submit our data to DEQ. And so DEQ has used our data in a lot of different ways, including the development of TMDLs. You've probably heard that term a lot if you've been tuning into the series, but just in case you haven't heard it yet, TMDL stands for total maximum daily load. And it's basically the maximum amount of a pollutant that a water body can handle or receive while still meeting its water quality standards. And that term is used in plans to restore waterways and remove them from that list of impaired water. So it's basically like a pollution cleanup plan. And we've contributed really essential data to a lot of different TMDL processes in the Rivanna River watershed, including the North and South Fork TMDLs. And I think with that, I'm gonna go ahead and turn over to Nesha, who's gonna explain a little bit more about how our monitoring data fits into DEQ's purposes and then some of the monitoring that they do themselves in the county.
Great, thanks so much, Rachel. I'll start. I think you have to stop sharing your screen for me to share mine.
Okay, thank you for bearing with me. I appreciate it.
Oh, no, you're fine. Okay, let's see. Here we go. All right. Well, thanks so much to everybody at Albemarle County for hosting this webinar today and for inviting me to be a part of it. I'm really excited to tag team a presentation with Rachel. I thought that was a great, really informative overview of all the great work that RCA does. So thanks for that. My name's Nesha McCrae. I work for the Virginia Department of Environmental Quality in our Valley Regional Office. I spend most of my time studying our waterways and working pretty closely with communities to try and better understand how we're impacting stream health and the things that we can do to improve it. So my plan for today was to give you all an overview of the work that DEQ does to assess the health of our state's waters, and specifically talk about Albemarle County, and explain how we apply what we learn through our monitoring efforts to help make our waterways healthier and to make them more resilient. So start with a big question here. How do we know if our streams are healthy? The more data we have to answer this question, the better. We can look at stream habitat, water chemistry, all of those bugs that Rachel talked about that are just fantastic indicators of all sorts of things that are going on in the stream. So we want to collect as much information as we can, and then we've got to look at that data and try and figure out what it's telling us. So DEQ conducts a water quality assessment of all of the waters in the state every even-numbered year. So we're looking at every 2 years. So our most recent assessment was done back in 2020. And we look at, I think Rachel said they look at a 3-year window. We look at a 6-year data window. So 2020 looked at 2013 through 2018. And so we conduct this assessment. We create a report showing all of the results of monitoring across the state. And we use this report as an opportunity not only to identify those streams that look good, that look healthy, but also those where we're seeing some problems. And it's those problem streams, what we call impaired streams, that we need to take a closer look at and prioritize for what we call cleanup plans. So I wanted to definitely mention where citizen monitoring data fits into our process. Rachel touched on it a bit in her presentation. And, not even talking about the relationships that these monitoring organizations build in our communities and how that helps us. That's a talk in and of itself. Just looking at the numbers. So this is a snapshot from our 2020 assessment. We had about 3,500 DEQ monitoring stations that we used in that report. In addition, we had over 1,500 citizen monitoring stations that were incorporated into this report. So we've got citizen monitoring data approaching that 50% mark, coming close to it for our water quality assessment. So that's really fantastic. It speaks volumes towards these organizations. And to hone in on things a little bit further, so DEQ has 6 regional offices across the state. So Albemarle falls in our Valley region, which is a 14-county area. So our portion of the assessment for 2020, we had 978 DEQ stations. And then we had 306 citizen monitoring stations used in that assessment. Just about half of these came from RCA. So when you put that into perspective, think about that for a minute. We've got a 14-county area where just about half, a little over half of the citizen monitoring data came from one county collected by one organization. So that really speaks very highly to the great work that RCA does. So I've mentioned monitoring and assessment. So what are we looking for when we monitor? What are we assessing? So in Virginia, we've got a series of what we call designated uses for our waterways. And this is a list of them. And so basically here, what we're saying is that our water bodies should be able to be used for a series of different purposes. So for recreation, it should be safe to swim in our rivers and streams without worrying about illness or infection. We maintain that our public water supplies should be free of any toxic chemicals that would make our water unsafe to drink. Same goes for wildlife, that our streams and our rivers and lakes should support a healthy population of wildlife, that there shouldn't be contaminants in them that would hurt wildlife. You should be able to harvest shellfish, things like mussels, oysters, clams from our rivers and streams. And you should be able to eat the fish that you catch. You shouldn't worry about mercury and PCBs in fish tissue. They should be free of those contaminants. And we say that all of our streams should support a healthy and diverse population of aquatic life. That's at Aquatic Life Use. So I don't have a formal poll for anybody, but just take a minute and in your own minds right now, think what 2 uses on this list here do you think we have to work with the most and end up studying the most, where we find we're not supporting those uses? So I'm going to pause, think on that for a minute. So here are our culprits that we deal with the most. The first is recreation. And so we're assessing whether we're meeting that use or not by looking at E. coli concentrations in the water. E. coli is a really great indicator organism. It tells us that there might be other things in the water that could make you sick if you get it in your eyes or your mouth, things like Giardia, Cryptosporidium. So we struggle with that. And then this aquatic life use. So that's where we're looking at those bugs that Rachel talked so wonderfully about with us. So those are the 2 things I want to focus on for the remainder of the time that I have. So let's take a look at this aquatic life use where we're monitoring bugs. So this is a map of Albemarle County, and I'll start by pointing out those purple dots. So those purple dots are showing the location of DEQ monitoring stations where we collected bugs, and we also looked at habitat between 2013 and 2018. So the first thing I'll go ahead and point out, because I'm sure everybody's thinking it right now, is, gosh, those all look to be clustered on the northern end of Albemarle County. And that is true. And the other thing you'll notice that's clustered on that northern end are the red lines. And so those are those stream segments that we said are not supporting that aquatic life use, where the bugs don't look so great. And so when we find those impaired uses, when we find that the bug population doesn't look great, that does really influence where we're going to continue monitoring. So when we know there's something wrong, we need to monitor it more to better understand what's causing that problem. So that does affect where we focus our monitoring efforts. And so you all may be thinking, well, that's great to study the streams that aren't doing well, but what about the ones that are, those green lines there that are fully supporting that aquatic life use? And, the other key driver in our Benthic monitoring program is what we call our probabilistic monitoring program. And this program's been around for just about 20 years now at DEQ, but it's really grown, and it's really a fantastic source of information when it comes to real trends that we're seeing in stream health across the state. And the thing that makes it so useful is that our monitoring sites in that program are selected randomly. So we get a real unbiased sampling of stream health across the state. And our monitoring through that program is pretty extensive. We're looking at bugs, habitat, physical, and chemical parameters in streams. So that really helps us get at some of those headwater streams and some of those high quality streams as well. But getting back to this map, you can see I mentioned the red line. So we're at 9% there for Albemarle County where we're not supporting that aquatic life use. But the good news is just about over half of that, again, is fully supporting. So we're at 17% showing those green lines. And about 74% we consider to be not assessed. Those are those blue lines. So that means that between 2013 and 2018, we didn't have data for those streams to draw a conclusion as to whether or not they're supporting this use. We'd certainly like that number to be higher. We're dealing with limited resources to get at all of those blue lines that you see there. And another thing to keep in mind with biological monitoring, you can't sample just anywhere. You've got to find a good riffle. You've got to have access to it, so you deal with private property issues. While all of our monitors love to go on a good hike, they only have so much time in a day, so you've got to find a place that you can get to in a reasonable amount of time. So you've got to have roads nearby. And we also really try and avoid sampling directly downstream from impoundments, things like big lakes and ponds, because those aren't really free-flowing systems. So that can complicate things as well. And I will note, we have a process where citizens can nominate streams for monitoring by DEQ. And right now that process is open for nominations through April 30th. So I'll send out a link to that location on our webpage through the chat box when I'm done presenting. But that's another way to add on to where these purple dots are located. And so this is a similar map, but we're going back to that recreational use assessment where we're looking at E. coli concentrations in the water. And so the orange dots you see here show DEQ ambient monitoring locations. So these are points where we're collecting a water sample and we're looking at different chemical parameters in the water, including E. coli concentrations. And so you can see a similar trend here, about 9% not supporting, those red lines you see, about 7% fully supporting, and about 83% considered to be not assessed. Those are those blue lines. Again, we want to get to more streams than we are now, but we do the best we can. With these ambient water quality stations, we try and maximize our resources by rotating off of stations every 2 years. So we'll typically visit a site every other month and do that for a 2-year period and then move off of that station to try and get better coverage across the state. We also have some long-term trend stations, which we've been monitoring for many, many years, and those really inform us as to some of those more temporal trends, things happening over time, big changes that we see in our streams. So a question that I get a lot from people is, how does Albemarle County, or how does my community compare to the rest of Virginia? So this table is an attempt to show you all that today. So this is data taken from our 2020 assessment, covering all of the potential water quality impairments. So not just bacteria, E. coli, or the bugs, looking at everything. So you can see Albemarle County stacks up pretty well against the rest of the state. You know, I think the numbers are pretty comparable, but a little bit better in most instances. And when you crunch all of the numbers of everything that was assessed, so when you take out that non-assessed portion, you're looking at a little over 50% of streams in Albemarle County having some sort of water quality impairment. So there's certainly room for improvement there. So I've talked a lot about how we monitor and how we assess, but what do we do about those red lines? What do we do when we see that a stream is unhealthy? So this is our water wheel, and it's all revolving around those water quality standards that are designed to protect those uses, things like being able to swim in streams without worrying about getting sick. So you start at the top, you monitor, you assess, we report, and then we need to study those streams that are appearing to be unhealthy based on our assessment. And so we do that through water quality studies called total maximum daily loads, or TMDLs. And I think Rachel provided a great overview of that. I could give a very lengthy talk on how those work, but I think that the main thing to note in those studies is that they spell out where pollution is coming from, you know, what particular pollutants we should be concerned about in a stream, and the types of reductions that need to be made to those different sources of pollution in a watershed to restore a stream. So once we do those studies, then we develop plans to implement them, to implement those goals, and then we monitor to make sure that we're reaching those targets. So it's a process that goes round and round, and that's a good thing. We want to continue to monitor and continue to make progress. So this map shows you where we've done these water quality studies, these TMDLs in Albemarle County. So you can see the Hardware River down there in green, along with a portion of the James River, the Slate River watershed in yellow, Moores Creek, and the Rivanna River. We've completed studies looking at excess E. coli concentrations in those watersheds. And for the Hardware River and Moores Creek, I've got them underlined because we've also developed implementation plans for those watersheds and supported some implementation efforts through different funding initiatives in those areas. We've looked at aquatic life impairments in a number of watersheds in and around the Rivanna River and Moores Creek and several tributaries around Charlottesville. And we found a lot of issues with sediment in those studies. So that's been a real focus of those aquatic life impairments in Albemarle County. We've also seen some issues with phosphorus in a couple tributaries of the North Fork of the Rivanna River, Blue Run and Stanardsville Run. And I wanted to note the North Fork of the Rivanna, we're in the process right now of we're planning to plan. We're working on plans to start up an implementation planning process there. And then the other thing I wanted to point out are these watersheds you see highlighted in pink here. That's the South Fork Rivanna River watershed. And we're working on a study right now in that watershed. And we've got a technical advisory committee in place. And if anybody is interested in learning more about that effort, please reach out to me. We'd welcome additional community involvement in that process. But right now, all signs are pointing to sediment as a stressor to aquatic life in the South Fork Rivanna, along with some nutrient issues in a few of those streams. So please reach out if you're interested in getting involved in that effort. So I wanted to wrap up with just a couple of kind of key takeaway points about the health of Albemarle County's waterways. As I think many of you probably know already, we're seeing the highest impacts to stream health in those urbanized areas in and around Charlottesville. And I think we know the more pavement you lay down, the more problems you usually see in our streams. Sediment is really the predominant stressor to aquatic life that we're seeing in Albemarle County, although we're also seeing some issues with phosphorus, which makes sense because phosphorus binds really nicely to sediment. However, I think Rachel's summary of their stream health report shows you a lot of those biological impairments are kind of marginal. They're approaching that threshold of that score of 60 that you're aiming for. And we see the same thing with these recreational impairments. E. coli concentrations are not, way up in the thousands like we see in some streams. They're really borderline too. And what this all really means is that there's really high restoration potential in Albemarle County, particularly with these great citizen groups you have out there monitoring and the level of interest and engagement that you see in Albemarle County. So I think there's some real promise there, and that's a really good point to end on. So with that, I'll turn things back over to folks with the county who wanted to handle any Q&A. So I will stop sharing here. Or do you all just want me to leave this slide up?
Oh, you can just leave it up for now while we take the questions. Thanks, Nesha and Rachel. That was great and very informative. We have a couple of questions coming in, and anyone else out there, if you have questions, go ahead and put them in the Q&A. We have definitely a little bit of time left to take those. So I'll just go ahead and read out the questions so that folks can hear them. Nesha, I think this one is for you. How much is DEQ using the results of RCA testing in its reports?
That's a great question. And sorry, I've pulled up that DEQ page there for you all to see. I'm going to go back here. I'm having trouble getting back. There we go. Sorry. Yes. So question. Yeah, RCA data is a huge part of our assessment. Since they're at that level 3, that means that we use it the same way we use our own data. And honestly, you know, that focus that we've had, where you saw all that biological monitoring and all those red lines clustered in the Rivanna River watershed, you know, the work that they're doing right now has really driven our focus when it comes to our water quality studies and our TMDL process. So, yeah, boy, they've been a fantastic partner and had a huge role in our assessment.
Great, thanks, Nesha. So another question, for the long-term RCA data, were effect sizes, correlation strength also calculated as part of the statistical analysis? I did not see them in the report.
We have a sum of correlation coefficients, and I'm happy to provide that information to whoever asked that question.
Great, thanks, Rachel. Another question here from Martha. It says, I wonder how the increased use of outdoor spaces will affect the water quality. Do you have any preliminary data from 2020 showing increased impairment?
I'm happy to speak to that, or I don't know if Rachel wanted to chime in, but you know, I would say the benefits probably outweigh the potential negative impacts. You know, I think for people to get invested in participating in water quality improvement efforts, getting them out on our waterways is one of the most effective ways we can do that, because they've got to value what they have, and they've got to think that it's worth saving. So, you know, I could see some instances maybe of additional pollution, probably mostly in the form of litter. But no, I haven't seen, you know, in looking at kind of our preliminary data, I haven't looked, our 2020 biological monitoring data hasn't all been processed yet. So I don't have any good bug data yet for that. But yeah, I would expect, you know, minimal negative impacts from that.
We've got this is more of a comment. I think it says, this presentation was quite good and understandable for the layperson. The graphics were great. Nice that the program was being made available for the hearing impaired. Thanks to all who were involved. Thanks, Kent and Judy, for that comment. And if there's any other questions, please go ahead and put them in the Q&A at this time. I'm not seeing any more. We'll give a moment here. But thank you again to Rachel and Nesha for presenting. This was just wonderful, really great to have you here today. And I hope that, you know, you all will come and join us next week. We're going to have Piedmont Environmental Council here talking about their work with HOAs and how they're managing common spaces in HOAs to improve water quality and watershed health. So thank you all for joining us, and please come back next week.